Good afternoon. We'll go ahead and get started. My name is Jeremy Still, and I have the honor of participating in today's webinar with Dr. Zhiying Li. So welcome to today's webinar. It's a rapid NASH model and its application into candidate drug screening for metabolic syndrome and hepatic steatosis, presented by Dr. Zhiying Li. Dr. Li is the Director of Life Science and Technology at GemPharmatech. Dr. Li obtained her PhD in neuroscience from Harvard University, the Department of Molecular and Cell Biology, under the supervision of Dr. Venkatesh and Murthy. Afterwards, she worked with Dr. Jeffrey Friedman for her postdoc training at Rockefeller University, studying the neuro and endocrine regulation of food intake and metabolism, including obesity and diabetes, in the context of leptin signaling. Today, she is going to share some very recent development at GemPharmatech in support of research in metabolism and liver diseases, in particular NASH. Dr. Li, the floor is yours.
Thanks, Jeremy. Hi, everyone. I hope you can hear me clearly, and very happy to talk with you and share some data with you. I'm gonna go straight to my screen share. All right, we'll get started. Well, thanks. So today I'm going to talk about NASH and also the mouse models and services that we have developed very recently at GemPharmatech. And if you were here with us in February, this is a continuation of the same topic from February. So hopefully today I will be able to give you some more new data and insights on how we're trying to help or advance researchers in the field of metabolism and liver diseases. So a quick disclaimer from the company. And so today, as I said, we're going to talk about NASH and NAFLD, the non-alcoholic fatty liver disease, NAFLD, as we call the early phase of NASH. So here is a slide on the background of the disease in the human population. Overall, worldwide, we have about 25% or 24% of the general public who has the fatty liver disease or early stages of the NASH disease. And as this disease progresses, it goes through the NASH, fibrosis, and cirrhosis stages, and eventually it can develop into hepatic carcinoma. At that stage, liver transplantation is typically required in order for the patient to survive. And in this disease, from NAFLD to NASH, there has been a lot of risk factors that are identified. For example, lifestyle, in particular, high calorie intake, as well as a sedentary lifestyle, obesity, diabetes, insulin resistance, as well as genetic factors. Aging also play a role in developing liver diseases in this aspect. And here on the right side is a cartoonish illustration of how the progress of this disease in particular can affect or alter the morphology of the liver cells. So we start with very beautiful, well-aligned cells in the liver. And once the fat accumulation starts, it's going to accumulate in the cell. And then once the accumulation increases, it will start to trigger inflammation in the cell, which leads to immune cell infiltration, as well as fibrosis, ballooning of the cell. Eventually, there will be apoptosis, necrosis, as well as fibrosis that are accumulating in the liver. So, as I said, this is a prevalence, not just in the States or in Asia, it's worldwide. It's associated with both genetic factors as well as environmental, lifestyle, metabolic factors. So, GemPharmatech, as we develop our commercial pipeline, we are a mouse model provider in terms of human diseases. We would like to assist researchers in different disease areas to use the best mouse model in supporting their preclinical research. So we position ourselves at the preclinical stage. We would like to use mice as a way to recapitulate the human disease so that research can be carried out in order to develop medicine for the human disease. So as an intro to the human situation here, I'm listing out a few factors that when we look at human disease or human NASH, what is typically looked for in the patients? Obesity, hyperglycemia, hyperlipidemia, insulin resistance, as you can tell, as well as some liver pathology data, in particular, steatosis, inflammation, hepatic injury, which show up as a ballooning, for example, this big, inflated cells. And finally, the ballooning leads to cell damage, which will show up as fibrosis in the liver. So these are, instead of the cartoons in the first slide I showed you, these are real patient biopsies where these morphologic features are very indicative for NASH. So NASH is coined as a non-alcoholic fatty liver disease. However, with the past decades or a few decades of research and study, people realize metabolic risk factors are actually tightly correlated with the development and progression of the disease. So last year, 2020, there has been a recommendation, instead of calling this particular disease and the progression of the disease as NASH and NAFLD, NIH recommend that we change it to MAFLD and MASH. M basically means metabolic or metabolism associated fatty liver disease. So in this cartoon on the left side, as well as a documentation type of workflow, this is how metabolism or metabolic dysfunction has been associated with the development of this particular liver phenotype. So in terms of providing mouse models to support the research in this area, these are some classical models that you probably have seen on the market from different mouse model providers. So on the top bar, I'm showing you, typically we are using diet treatment as well as some chemical insultation to injure the liver in order to induce the phenotypic pathological changes that we saw with the patients. So these include steatosis, inflammation, ballooning, fibrosis. At the same time, given the knowledge that metabolism or metabolic dysfunction has been playing a key role in the onset and development of this disease, we are also looking into, the phenotype of obesity, glycemia, hyperinsulinemia, or lipid dysfunction in the models that we develop. So among all the classical models I have listed here, what we can see is with some diet treatment and chemical insult, one of the issues is that they don't really have the metabolic dysfunction. And then the high-fat diet and Western diet treated animals, they do have the metabolic dysfunction, and they're showing mild or appropriate liver histopathology, but at the same time, the timeline of the model where the disease progression has been so long, it is not really suitable for today's drug development requirements. So as I highlight here, so what can we do to overcome this shortcomings? So what we thought about is we would like to combine genetic dysregulation with a metabolic treatment, which is the diet treatment. So for today's topic, what I want to introduce to you as a continuation from the February webinar is the Alms1 mutant animal. So Alms1 is a gene that is responsible for a particular human inheritable disease called the Alström syndrome. This syndrome is present in the human population. They do have quite some metabolic dysfunctions phenotypes, including obesity, diabetes, vision problems, sensory deficits, cardiovascular problems, reproduction defects, as well as renal dysfunction. The function of this particular gene, the AMS1, is not fully understood at the moment. However, it is found to be expressed highly in the villi of the intestinal cells. At the same time, it seemed to be playing a function at the centromere of the molecular machinery in the cell. So what is also known is among the human population which exhibits Alström syndrome, exon 8, 10, and 16 are actually hotspots where mutations has been observed among this particular patient population. So in our hands, we developed the B6-Alms1 deletion mutant animal by using CRISPR-Cas9. We deleted 11 base pairs in the exon 8 of the mouse Alms1 gene. And this created a frame shift mutation so that the mouse is actually having a loss of function phenotype due to this deletion in the mouse Alms1 gene. So here is a summary. In the next few slides, I'm going to summarize the metabolic dysfunction that we have seen in this mouse when they are treated or when they are fed with control normal chow diet. So as we can see, the black are the black animals, the B6 animals. The red is the mutant animal with the 11 base pair deletion in exon 8. So they have accelerated body weight increase, which represents obesity. They have higher blood glucose as well as higher insulin. And in this one, we did glucose intolerance and insulin intolerance tests. They are intolerant in both cases. And they have dyslipidemia as shown by higher cholesterol, triglyceride, HDL, and LDL in their blood. At the same time, they have accumulation of fat in the liver, as shown in this panel, as well as increased total triglyceride in the liver. Their liver function is also impaired, as shown by the ALT and AST tests. Now, we challenged the animal with a Western diet. So Western diet is different from the high-fat diet in a sense. High-fat diet is only high in the fat content of the diet. Western diet is also is not only high in the fat, but also in glucose. So is a more similar to the so-called Western diet, and with a highly processed food as well. So treating the animal with a high-fat diet, which is shown by the blue bars or the blue color, as you can tell, as the animal grow older, treated with high-fat Western diet actually accelerates their obesity or their body weight gain. It also accelerates their accumulation of the triglyceride, cholesterol, LDL, HDL in the blood. Their ALT and AST level is similar to the mutant animal on the chow diet. At the same time, they do have a bigger liver, and their liver is having a lot more fat on average than just a mutation or than just a mutant by itself. In the next one, this is the most relevant for the disease onset and progression. So not only the animal need to have the metabolic dysfunction, but also the liver pathology or histopathology is a hallmark of the onset and progression from NAFLD to NASH. So here, the NAFLD or the NASH is scored by a standard scoring system, which is called the NAFLD activity score. It basically evaluates the liver histology or morphology by the content of steatosis, the amount of inflammation, and the amount of ballooning that can be observed from the tissue. So as you can tell from here, the B6 animal on the chow diet, the mutant animal on the chow diet, or the mutant animal on the Western diet. As we can tell, this is the HSE, basically showing steatosis, the accumulation of fat, the ballooning as the big, inflated cells are trying to show, and also the blue area here is trying to show the infiltration of immune cells or inflammatory cells in the tissue. The red oil staining is basically showing one more time there's a lot more fat accumulation in the mutant animal eating the Western diet. And finally, the fibrosis is shown in the bottom panel. This mutant animal with the Western diet is having a lot more profound fibrosis, which is a hallmark of NAFLD developing into NASH. So there's one point I want to highlight, which is the mutant animal on chow, they are 21 weeks of age. The mutant animal on the Western diet, they're only 14 weeks of age and have been treated or has been fed on the Western diet for 6 weeks. So as you can tell, the disease progression is a lot faster and profound in this particular mouse. So we do think we have found a new model that we can mimic the NASH situation or the disease progression, and as a way to recapitulate the human NASH situation. So basically, I'm summarizing here. This animal with the Alms1 loss of function mutation on the Western diet treatment, we do think it mimics NASH and can be used as a mouse model for the study or drug testing when we want to develop or assist the development of human medicine. So this is our service outline, where we do want to provide testing services using this well-established mouse model with the diet treatment. So in our hands, we outlined the service platform as in this illustration. So animals are 6 weeks of age when they are randomized to be segregated into the drug treatment group versus the dosing, the actual testing article group. And then within this 8 week of treatment, we're gonna take blood samples at midpoint as well as endpoint. And then also at the endpoint, we're gonna take tissue. We're gonna take liver biopsy to do histology, as well as any other custom analysis measures that that our customers would like to do. I want to point out, I misspoke for this 6 week. So the 6 week here indicates the Western diet treatment. So typically the mice are 8 weeks of age when they were put on a Western diet, and then having 6 weeks of Western diet treatment before they're tested either with vehicle or the actual testing article. And then the experiments goes on as outlined here. So in this one, as a test of, as a proof of principle, we took advantage of 2 very promising phase 2 to phase 3 clinical trial article, which is the MGL 3186 and the semaglutide. So these 2 drugs were developed for 2 different indications. One is in the GLP-1 receptor inhibition. The other one is the tyrosine thyroid hormone receptor beta pathway. So they were meant to regulate thyroid pathway as well as the insulin resistant pathway. However, with new clinical trials, they have been showing promising data in the treatment of NASH. So we want to see whether the Alms1 mutant animal fed on a Western diet, whether these animals can respond and report the clinical response profile as we have seen among the human population. So here, let me draw your attention to the different coloring. The black is the B6 animal. The red is the our NASH model, basically the mutant Alms1 with the Western diet treatment. And then the blue and the gold or the brown color, they represent the NASH animal treated with the MGL3196 or the semaglutide. As you know, the dosing arrangements are illustrated here. So as you see from these two graphs, when the animals are given these two drugs, they actually were able to respond by losing body weight or lowering the increase of their body weight. At the same time, here looking at liver weight, the two drugs can lower their liver weight, and mostly that's, as you can see from the next couple slides, that's the reduction of fat content in the liver. At the same time, having the drug treatment can also reduce the fat weight in the white fat, different fat depots. So this is the sub-Q white fat, this is the mesenteric white fat, as well as the epididymal white fat. And then surprisingly, in the interscapular brown fat, there's no difference between the drug-treated versus the untreated animal. At the same time, this is the lipid profile. So with the drug treatment, you can tell, or you can see from this this plots, both of them are effective in terms of reducing the total fat accumulation in the liver. And this is the liver function. So the two drugs were able to restore partially the liver function in these animals. And finally, as you know, as we say, the histology is really the one of the hallmarks for this NASH symptom. So here, the B6 animal treated with, well, they're fed on the chow, treated with a vehicle, versus the mutant animal treating fed on the Western diet with a vehicle, and then the mutant animal on the Western diet treated with the two drugs. Morphologically, you can tell, you know, the two drugs were able to reduce the steatosis as well as the ballooning in the tissue. That's very obvious. And then when we do the NAFLD score, activity score, the different colors are indicating different aspects of the NASH. So the pink at the bottom is steatosis. The purple blue is for ballooning. And then the red on the top is for inflammation. So both of the two drugs were effective in terms of reducing the NASH phenotype in these animals. And then finally, this two panels shows the reduction of total liver triglyceride and cholesterol in these animals. And then finally, another thing, fibrosis is really one of the irreversible phenotypes. Once the NASH has developed fibrosis in the liver, reverse the fibrosis is really one of the hardest things that we're trying to develop drugs against. So in these two cases, having these two drugs treating the NASH mice, we're seeing a slight reduction in the fibrosis, which is, as you can tell. So here, this three hotspots illustrates quite a bit of fibrosis going on and is alleviated to some extent, although not completely, by the two drugs in the mice. So in conclusion, I hope the data I've presented or shared with you today illustrates to you that the Alms1 mutant animal, they do have metabolic dysfunction and they do develop fatty liver due to the genetic mutation. At the same time, a Western diet feeding regime will accelerate their metabolic dysfunction as well as prompt them to develop NASH or NAFLD until NASH in a very fast manner within 6 weeks. in our hands or in our standard treatment procedure. At the same time, though, this particular mouse model with the Western diet treatment, we think is a good model to mimic the human NASH disease stage, and is a promising new testing platform for new NASH therapeutics. So as a proof of principle, we have seen they can respond, and they are quite sensitive to the two promising drugs that has been gaining really momentum in clinical testing. So that concludes for the data on the NASH models or on the Alms1 mutant animal with the Western diet as a NASH testing platform. I also want to give a shout out to this particular drug screening project. So at GemPharmatech, we're trying to develop a lot more humanized animals as well as mutant animals that can support drug screening for particular human diseases. So here, I don't want to belabor you by reading all these gene names, but we have quite some that in green, they're available already in our on the shelf products. We have quite some that are illustrated in blue that are upcoming next year, somewhere in the middle of the year. So before I close, I do want to give a update. Our next webinar is not going to be the end of November, but rather at the beginning of December. This is due to the holiday, the Thanksgiving holiday that is coming. And near the end of December, that's going to be Christmas. So we'll have a delay in the December as well. If I'm not mistaken, the one after December will be 2022 January. So I want to remind everybody, if you are interested in rare diseases, we do have a funding opportunity that we support 85% of the cost associated with the mouse model development or construction for a rare disease. If you have an idea, we are here to support you, and we would like to create a mouse model for you in your study. And at the same time, as you are aware, GemPharmatech is growing. So we are hiring new people in these four particular areas: for our client manager, our R&D support scientists, as well as business development personnel. And we also are hiring for scientific content creator. And this is my last slide. Has our contact information, and feel free to browse our website, gempharmatech.us, to get more information about our company and products and services. And thanks a lot for your attention. And if you have questions, I'm happy to answer.
Thank you, Dr. Lee, for such an informative presentation on NASH models. We do have one question so far. The question is, are these models available in the US?
At the moment, the mice can be available in the US. However, the service associated with the mice are not available in the US. They are available from our parent company in China.
I'm not seeing any additional questions at this time, but we can just give it a moment. If anyone wants to actually ask a question, go ahead and in the bottom of your screen, you can hit the Q&A button to ask anything you like.
You know, actually, I have a question for our audience. How many of you are actually doing, NASH research or research on liver diseases? If you don't mind, you can share that with us in the chat box.
I'm not seeing any comments. Oh, we do have one question that just popped up. Do you do testing for clients, or do you send out gem mice?
Well, as I said, at the moment, we provide, in the US, we can provide mice. We don't have the service capacity, but in our parent company, we provide mice and we do the service for clients in-house.
I'm not seeing anything else popping up in the chat or the questions.
Well, in that case, I think I'll just say thank you everybody for coming. And if you have questions later, you can always reach out to us. You know, or by visiting our website. All right, thank you.
Again, thanks, Dr. Lee, and thanks to everyone for joining us today. And we look forward to seeing you in December.
Bye.
Bye-bye.